first talk is from Junior West, who's going to tell us about an Arf-GEF called STEP that antagonizes actomyosin activity.
Great, thank you. And thank you to all the organizers of the conference for giving me the opportunity to come here today and share some of my work with all of you. So in our lab, we're interested in studying actomyosin networks. And the formation of these actomyosin networks are important for driving various forms of morphogenesis. However, in our lab, we're interested in studying genetic mechanisms that are important for holding back and antagonizing these actomyosin networks. And we became interested in this when a previous PhD student by the name of Hoon was studying the Arf-GEF Stepkey, and he found that Stepkey actually antagonizes actomyosin networks. Now, Stepkey is a member of the conserved family of Arf-GEFs, and Hoon was studying Stepkey during the early cleavages of the Drosophila embryo. Now, during this stage, furrows from the plasma membrane come in to separate individual nuclei to form cells. And at the base of these nuclei, there is an enrichment of actomyosin networks. Now, when Hoon depleted these embryos of Stepkey, he saw that there was a drastic expansion of these actomyosin networks that led to various disruptions during embryogenesis, such as premature cell division. So for my project, I became interested in trying to understand if Stepkey played a role during later stages of embryogenesis. Now, the precedent is already there because we know that actomyosin networks are critical throughout all stages of embryogenesis, and in particular during these late stages, such as dorsal closure, when cells undergo this elongation process in order for tissues to spread over the entire embryo. Now, during this process, we see that actomyosin networks are enriched at these elongating contacts, which is kind of counterintuitive and suggests that there's some kind of need for regulated contractility. So in order to see if Stepkey is involved in any of these processes, the first thing I did was to look at zygotic loss-of-function mutants for Stepkey. So when I did this, the first thing that I observed was that these mutants were embryonic lethal, so they didn't hatch as larvae. And when I looked at the cuticle of these embryos, I noticed that there were head holes in the cuticle. So the next thing I wanted to do was to see if I could see where these defects first began during embryogenesis. And to do that, I decided to do some live imaging of embryos expressing this DE-cadherin GFP construct in a wild-type or a mutant background. So in wild-type embryos, and this led me to look at the onset of dorsal closure. So during this process in wild-type embryos, we see that the epidermis normally takes on, or the cells of the epidermis normally take on this regular cobblestone array. And as dorsal closure begins, these cells elongate and take on this rectangular array. So when I looked at my Stepkey mutant embryos, I observed that there are patches of cells that had difficulty in establishing this process and did not take on these uniform rectangular arrays. So I also fixed and stained some embryos for DE-cadherin at later stages of dorsal closure. And when I did this, I observed similar defects. So notably, we see here at the leading edge that in the control embryos, we notice that the leading edge has this uniform cell length at this contact right here. And in the mutants, we observe that this uniformity is lost, and we see more variability in the edge lengths. In the controls, we see that the cells take on this regular rectangular array, as I mentioned previously. And we see in the mutants that there are cells, just here, that do have, again, take on these irregular cell shapes. And we also notice that the formation of ectopic rosette structures that we define as these 5 or more cells that come together at a single point. So we can measure and quantify all of these characteristics, and when I did this, I noticed that there was a significant difference between control and mutant embryos. So given what we know about the relationship between Stepkey and actomyosin during early stages of embryogenesis, I wanted to look at these networks during later stages. So to do that, I looked at these embryos expressing this myosin heavy chain, or zipper GFP, and I fixed them and stained them with phalloidin to look at the F-actin networks. And actomyosin networks are very well studied during this stage of embryogenesis, and my results were consistent with this. So in particular, we noticed that there is an enrichment of myosin at the leading edge, forming this uniform purse string. When we look at the apical side of these cells, we see that they form this uniform apical cap. And if we just scan down one section to look at the junctions, we see that the formation of these myosin bands that run along the dorsoventral axis. When we look at my Stepkey mutants expressing this super GFP, we see that the leading edge seems to take on a less uniform appearance, where we have some areas where we have lower myosin and some areas where there's higher myosin. When we look at the apical side of all the cells, we see that the myosin seems to take on a more of a punctate distribution. And a lot of these accumulations of myosin seem to be localized at these tissue distortions. So specifically, when we look at multicellular rosettes, we see that myosin seems to be accumulated right at the center. So since it seems as though loss of Stepkey seems to lead to accumulation of myosin, we know that increases in myosin also lead to increased tissue tension. So we wanted to see if our Stepkey mutants also displayed some differences in tissue tension. So to do this, we collaborated with the Fernandez-Gonzalez lab, and how we decided to test this was to use a UV laser to make incisions in the epidermis. And much like when you cut an elastic band, the recoil away from the cut site is proportional to the amount of tension that's in the tissue. So for our experiment, we decided to cut circles in the epidermis to isolate patches of cells from the surrounding tissue. And what we're seeing here, these are actually holes. This circle that you see here are holes in the vinylin membrane that are left behind from the UV laser. And you can see that, as I mentioned, in control embryos, we see that the patch of cells does recoil from the initial cut site. So when we compare our control and Stepkey mutant embryos, right after ablation, we can see that in the mutants, we see a larger distance between the patch of cells that was isolated and the surrounding tissue, suggesting that there was more of a recoil in this first time point. We can actually measure the area of that patch of cells over time. And when we do that, we can see that the area right after ablation in our Stepkey mutants seems to reduce more than the control embryos. And we can then use these measurements to derive the speed of recoil. And when we do that, we see a significant increase in the amount or in the speed of recoil in our Stepkey mutants. So taken together, this all suggests that loss of Stepkey does lead to elevated tissue tension. So far, I've shown you that Stepkey seems to be antagonizing actomyosin networks. During early embryogenesis, that is controlling cytokinesis. And during later embryogenesis, we've seen that it's promoting tissue plasticity. So the next thing we want to know was, how does this actually work? So the first thing we decided to do was see where Stepkey is actually localized. And to do this, we fixed embryos at various stages of embryogenesis. So here I'm showing 2, stage 8 and stage 13. And we use an antibody that we obtained from the Hoke lab. And when we did this, we saw that Stepkey generally is localized to the plasma membrane and is specifically enriched in areas where we know there's high actomyosin activity. So specifically, we see high levels of Stepkey at cytokinetic rings of dividing cells, at these constricting cell-cell contacts, and along the leading edge. Here. So it seems as though Stepkey is acting where actomyosin is. So the next thing we wanted to know was to see if it's using its GEF activity. So to do this, we expressed 2 GFP Stepkey constructs, either a wild-type Stepkey construct tagged with GFP, or a Stepkey construct that is deficient in its GEF activity, so it's not able to activate downstream ARFs. And we're expressing these constructs in a Stepkey mutant background using this paired Gal4 driver to achieve this striped expression pattern. So when we do this, right away we observe that the wild-type construct alone seems to be rescuing the cell shape and the number of rosettes, as you can see by counting the number of rosettes and stripes that contain wild-type Stepkey. There's less than in the mutant one, in the stripes that lack this construct. And this seems to be dependent on the GEF activity. And we can also count and quantify these, and we see a significant difference in stripes that contain wild-type Stepkey versus stripes that don't. And again, we don't see that when we express the GEF-deficient construct. So this suggests that downstream ARF activation is critical for these effects. So one interesting thing that we noticed when we were imaging these embryos, specifically the GEF-deficient construct, was that the GEF-deficient construct seems to be drawn into the center of multicellular rosettes. And as I've told you, this is where we know there is high actomyosin activity or networks there. So this got us to thinking that perhaps actomyosin networks themselves actively recruit Stepkey. So to test this, we decided to look in mutants of myosin and Rho1, and we fixed embryos and stained them with a Stepkey antibody and specifically looked at areas where we know there's high Stepkey levels, so specifically at the leading edge here during dorsal closure. And in both myosin and myosin and Rho1 mutants, we saw that there was a decrease in Stepkey levels. In another experiment that I don't have time to share with you today, we also have seen that actomyosin is sufficient to recruit Stepkey. So taken together, what I've shown you today, so Stepkey seems to be antagonizing actomyosin networks. These same networks seem to be actively recruiting Stepkey, and together this forms a negative feedback loop. This negative feedback loop seems to be very local and acting locally at junctions to regulate them. And this local junctional regulation is critical for promoting tissue plasticity. So with that, I'd just like to thank my supervisor, Dr. Tony Harris, as well as my supervisory committee, Dr. Uli Tapas and Dr. Rodrigo Fernandez-Gonzalez. I want to give a quick shout out to Teresa in Rodrigo's lab for helping me with a lot of things when it came to numbers. And also all the past and present members of the Harris lab. And I just want to give two quick shout outs to Yishie and Francisco, who both have posters here this week. So if you're interested in other things that we do in our lab, please feel free to visit their posters. And thank you for your attention. I'd be happy to take any questions.
A really nice talk. I was wondering, your data is really interesting, and it seems like myosin 2 inactivation is required to elongate the junctions after rearrangements. So I was wondering if you see these oscillations between contracting and not stabilizing an elongating junction after rearrangement. It will be really consistent with what your data, so it's really interesting.
Yeah, so when I've imaged in wild-type embryos, when I image zipper, GFP, or myosin, I do see like increases and decreases in myosin levels. It doesn't seem to be very like coordinated. Like you don't see all of those junctions oscillating at the same time, but you definitely do see oscillation. I wouldn't say that it's as dramatic as oscillations that you see at other stages, so such as during germ band extension when you know there's oscillations in the apical medial area. But you do see it is a dynamic. These cables are dynamic, and that they increase and decrease in their formation. Yeah.
Okay, thank you.
Yeah.
So I understand STEP is an Arf-GEF, so is it known what's the molecular pathway by which it inhibits contractility? Is that known from the previous work?
Yeah, so it's not exact. So as of right now, we're not exactly sure how it's working. But so Stepkey is an Arf-GEF at the plasma membrane. So it does. So from my work, it seems to be activating Arfs there. We know from work in cell culture that Arfs are known for changing the cytoskeleton, and they do this in order to promote different trafficking routes in the cell. So we think that maybe somehow trafficking and specifically endocytosis at the plasma membrane might be somehow related to the antagonism of the contractility machinery. But at this point, we're not exactly sure how it's working. Yeah.
Any more questions? I'll ask a question. In your ablation experiments, have you looked at how actin, myosin rearrange, or in both your wild type and mutant?
Yeah, so no, we haven't imaged actin and myosin, no, in our ablation experiments. But that definitely would be something interesting to look at to see if we see a difference in the dynamics. I have looked in the amnioserosa, just under wild-type conditions, to see if there's difference in dynamics of myosin in the control and mutants, and we don't see anything too drastic. So I'm not sure but yeah, that's something that definitely would be interesting to look at. Yeah.
I may have missed this, but there are rosettes that form much earlier than the defects that you showed later. Is it obvious why the Stepkey phenotype is appearing in these late rosettes and not the earlier rosettes?
Yeah, it's a great question. So I guess the rosettes you're referring to are in germ band extension when we have this convergent extension happening. So the reason why, we're not exactly sure why they're happening specifically. I mean, we think for sure it's definitely linked to actomyosin. So during these earlier stages, we know that contraction of actin-myosin network at specific contacts are what drives the formation of these rosettes. And so we think that that's probably what's also happening in this case. I haven't imaged myosin specifically to see if that's what's leading to it, but we think that could be what's leading to these rosette formations.
So what's the reason that the phenotype manifests so late and there isn't a more dramatic phenotype in terms of the extension and elongation?
Yeah, so the mutants that I generate are zygotic mutants, so the mothers are heterozygous. So we think that the maternal supply probably is what's carrying it to these later stages. Yep.
Okay, thank you, Junior.
Thanks.